Cell respiration. By the end of this video, you should be able to distinguish between aerobic and anaerobic reactions and compare and contrast photosynthesis and cellular respiration in the terms of energy transformation. These are the things you will be able to do. This is an overview of cellular respiration. It happens in both the cytoplasm and the mitochondria, and the main purpose of cellular respiration is to break down sugar molecules and produce energy. So, the process that releases energy by breaking down glucose in the presence of oxygen is cellular respiration, and this is the formula for it. This formula will be very important. 6O2 plus C6H12O6 yields 6CO2 plus 6H2O plus energy, or ATP. We break that down into words, and it just means oxygen and glucose yields carbon dioxide, water, and energy. Cellular respiration does require oxygen in a food molecule, and it will release carbon dioxide, water, and energy. So what we put in is oxygen and food, what we get out is carbon dioxide, water, and energy. It happens in a series of steps similar to photosynthesis, and the cell will use respiration to trap energy in the ATP. Glycolysis. This is where we break one glucose molecule in half, and we produce pyruvate, 2 pyruvates for every one glucose molecule. It will happen in the cytoplasm, and it is anaerobic. Anaerobic means that it occurs without oxygen, and it uses 2 ATP to get started. These are invested molecules, things that we put in to get the product out that we need. And we pass electrons on to NAD+, which is just an electron carrier. The glucose will enter the cytoplasm of the cell, and 2 ATP will start the process by adding in energy. Glucose will be split into 2 pyruvic acid molecules, and we form 2 NADH and 4 ATP. 2 ATP are created. Remember, we invested 2 to start the process, and we made 4, so our net creation of ATP was 2. Now, the pyruvate is ready to move on to the next cycle, which is the Krebs cycle, if oxygen is present. If we do not have oxygen, then we go through anaerobic respiration. So, if oxygen is present, aerobic. If oxygen is missing, anaerobic. An means without. Fermentation is the process that we undergo if we have no oxygen. This is where we ferment the pyruvate. It is not nearly as effective as cellular respiration. It produces less energy, and it will produce a little bit of ATP. Alcoholic fermentation is what's going to happen in bread or yeast to make it rise. Lactic acid fermentation happens in the muscles, so after a good workout, this is where your muscles will feel sore. It will occur in the absence of oxygen, and pyruvate acts as the final electron acceptor from NADH, producing lactic acid or alcohol. This results in no additional ATP production, and is used by many organisms in oxygen-poor environments, and it is used to regenerate NADH. So, lactic acid fermentation, just an overview. Pyruvate is converted to lactate. It occurs in muscle cells and is the cause for sore muscles. Bacteria is one of those things that will do lactic acid fermentation, like the bacteria that make yogurt. Alcoholic fermentation is where we convert pyruvate to alcohol and carbon dioxide, and it occurs in yeast. And this is an overview of that process of glycolysis. So, what is the chemical equation for cellular respiration? What type of process does this describe? And describe the process of glycolysis. How much energy is being made? Be specific. These are questions that you should be able to answer. And what is the difference between anaerobic respiration and aerobic respiration? Well, the chemical equation is glucose plus oxygen yields CO2 plus energy plus water. Glycolysis, we break down glucose into pyruvate. We make 2 ATP. Anaerobic and aerobic respiration. Anaerobic is without oxygen. Aerobic is with. Anaerobic does not make it nearly as much energy. Aerobic makes a lot of energy. So the Krebs cycle, after glycolysis where oxygen is present, pyruvate will move into the mitochondria, it's our powerhouse of the cell. It is the site of aerobic respiration, and the interior will consist of folded membranes, the cristae. This is where it will happen, and the inside of the membrane is the fluid matrix. The intramembrane compartment is between the inner and outer membrane, and most ATP production occurs in the matrix. The Krebs cycle, pyruvate is broken down and more energy is released. Electrons are picked up by carriers, NAD+, FAD, and ATP is also produced, just a little bit though. After the Krebs cycle, we move to the electron transport chain, and this is after 2 turns of the Krebs cycle because we had 2 copies of the pyruvate. All of the electron carriers will move on to the electron transport chain. They drop off their passengers, which would be your electrons and hydrogens, and these electrons go on a ride down the electron transport chain. And oxygen is the final electron acceptor. It will create water, and these electrons will help power the system that will make our ATP. As we have these electrons traveling down the chain, they are pushing hydrogen ions through the membrane, creating a concentration gradient. This buildup of hydrogen ions will help us make the ATP. At the very end of the process is ATP synthase, an enzyme, a protein that is powered by hydrogen ions. The ions are pumped through the protein, and the wheel is turned, and the phosphate will be joined to ADP, making ATP, and also making water. All of those words describe this picture. This is the electron transport chain. So our electron carriers, NADH and FADH, will bring their electrons and their hydrogens and drop them off. The electrons will ride down the chain, the hydrogens are pushed through the membrane, creating a concentration gradient. At the very end of the chain, hydrogens will join up with oxygen after being pushed through the ATP synthase. This ATP synthase will attach a phosphate to an ADP, creating our ATP. So, in summary, oxygen is the final electron acceptor and it makes water. Between 34 and 36 ATP are formed by one glucose molecule.